All right, for this second part of the lab, we're going to look at hydrogen specifically. And hydrogen, if you look at, this is the what we call so-called Balmer Series, which was discovered in 1883. And it's the only element, because hydrogen has one proton, one electron, that has a distinct pattern to the wavelengths and color. And so you have, in this case, we can see three, there's actually four visible ones, which we'll look at when we look at more detail. The red one, the blue-green one, and the dark violet. There's actually another one near the dark violet, which is almost impossible to see under normal conditions. It's very dim. So what we're going to do is look at this in more detail using the Vernier emission spectrometer and try and look at these lines much more precisely and measure the wavelength and compare that to the theory. So that is the rest of that will be what the experiment is, and I'll talk about it afterwards. For this part of the lab, we're going to be looking at the spectrum of hydrogen under more precise way of doing this with using the hydrogen tube. And what we're going to do is we have it hooked up to a fiber optic and then to a Vernier emissions spectrometer. And then what we're going to do is this will scan between 350 and 700 nanometers. And what we're going to do is look at the spectrum. So here we have the spectrum. And what I'm going to do is you've got the different lines. One here. So this is the first line over in the right, and we can get the actual, in the left here, bottom left, we have the wavelength in nanometers of this particular color. All right, and I'm going to move it over to the next one. So you have the first one. This will be the second one. And that is 486.6. And then we're gonna, because of the scale, all right, I want to try and get the last couple lines. So I'll change this to 0.2. So now we have the bigger lines, and you see we got this one. This is the third one. I will move the mouse to there. And that is at 434.3. And then we'll get the last line. I'll change this scale to 0.05. So we can see our last line. And this is our last line here. It's very hard, it's kind of dim. There it is, and that's at 410.3 nanometers. So these are the four lines of the Balmer series that are in the visible part of the spectrum. Physics 1102. We're looking at the first part of the lab was dealing with the gas spectrum. And that was to identify samples A through G of the different color, whether it's helium, different samples, and match those up. The second part of the lab, we're actually going to look at the hydrogen spectrum, H2 spectrum, which, as I indicated, showing you what the colors look like, is that the H2 spectrum has just one proton, one electron orbiting it, and as you can see from the lab, from the talking, is what we have is there are different orbits that are allowed, and this was discovered by Niels Bohr in 1913, over there. And what we see, these lines, as you just saw, for hydrogen, red, green, red, blue, green, and dark violet, come about from the electron when it drops from one level down to another one. So these are quantized orbits, much like planets, but they're quantized, meaning they can only be in distinct orbits. When it goes from one down, it emits a photon of light, depending on how high it's coming down to. And these orbits are n equals 1, 2, 3, 4, and so on, all the way out to infinity. And for the Balmer series, which is in the lab handout, is given the middle one where we have visible light. And that's when we drop down from higher levels down to n equals 2. If we drop down to n equals 1, that's going to be the Lyman series, which are UV, and it drops down to n equals 3. That's the Paschen series, which are in the infrared. So the visible light, which is, again, this was first observed in 1883 by Balmer. But he just came up with the pattern for it. And so what we're going to look at here are the values for the different colors. So there's going to be 4. I just showed you, we measured them. And I did the experiment using the Vernier emission spectrometer. And we're going to compare it to theory. So we have the wavelengths, the experiment using emission spectrometer. And we're going to compare this to theory, the wavelength in theory, using this formula. Of lambda is 1 over r times 1 over n sub f squared minus 1 over n sub initial squared, where n sub f, it drops down to 2, and n sub I is 3, 4, 5, and 6. Okay? And that corresponds to n is 3 is red, 4 is green, 5 is violet, which you can see. 6 is dark violet, which is very hard to see. And R is a constant, 1.0973 times 10 to the 7th. So that's how you do it. So in case it's so for the Balmer, we're going to compare, this will be 1 over 2 squared, which is 4, and then we would have for here, we either have 3 or 4 or 5 or 6, okay? And then this is R, so this is 1 over R, that gives us our wavelength in meters. And again, compare 1 meter, since we have 1 nanometer, is 10 to the minus 9th meters. So if you want to convert this to meter to nano, your answer, you're going to have to change that, move it over 9 decimal places. Okay, so that's the lab there. Where again, N sub F is 2 for all of these. And this will be 3, 4, 5, 6. We did that from the experiment. We get the theory by using the formula I've just explained here. And we'll get a % error for that. Okay, and I've explained all that. So that's for the hydrogen spectrum. Okay, so that's lab, and good luck with that.